Very good morning, dear students. Today I will be discussing about the biological classification, synopsis of it. So you can see in this slide diversity of life. So you can see there are vertebrates, mammals, birds, plants. So there is a huge diversity of life. So biological classification classifies all these organisms. So you would already have studied taxonomy. Taxonomy is a branch of biology which deals with the study of the characterization, classification, identification and nomenclature. So here biological classification, we are majorly focusing on the 5 kingdom classification, so which have been put forth. Now the primitive classification. See, Aristotle is father of biology, father of zoology. You know that Theophrastus is the father of botany. So Aristotle had a primitive classification. So he had his own limitation during those days when man had just come from Stone Age to the Greek civilization. So Aristotle was the earliest to attempt a scientific basis for classification of organisms. He classified plants in simple types like trees, shrubs and herbs. So these are the classification that he had. And he classified animals into 2 groups, those with red bloods and those without red bloods. So this is what is given in your book. Apart from that, if you see those with red blood, he included mammals, lizards, birds and fish. Those without red blood, he included hard-bodied, soft-bodied organisms. Hard-bodied included only insects. Soft-bodied with shell, without shell. With shell included shellfish. Without shell, jellyfish. So this was the primitive type of, very simple type of classification that Aristotle put forth. Now the 2 kingdom classification was put forth by Carolus Linnaeus, the father of taxonomy and father of binomial nomenclature. In 1758, he proposed the 2 kingdom classification. Last year, there was a question in NEET regarding the year the Montreal Protocol was signed and other things. So it's better that students focus on the years when these things were put forth as far as NCERT is concerned. So 2 kingdom classification was proposed by Linnaeus in 1758. So this system classifies organisms into 2 kingdoms. Plant kingdom, animal kingdom. It's a simple classification, only 2 kingdoms, plant and animal kingdom. But it had a lot of drawbacks. So the drawbacks of this 2 kingdom classification was prokaryotes without the nuclear membrane and nucleolus like bacteria, cyanobacteria, and eukaryotes with nucleus and nuclear membrane which included many members like fungi, mosses, ferns, gymnosperms and angiosperms. They were included under plants. So both prokaryotes and eukaryotes were included under plants based on the presence of only one feature, cell wall. But they were different in other features. Even chemically the cell walls were quite different. Linnaeus did not have access to know the biochemistry of the cell wall during those days. So this was one of the important drawbacks, not separating prokaryotes and eukaryotes, instead placing them under a single kingdom, plants. So it included, the second drawback is there was no differentiation between unicellular and multicellular organisms. So it included both unicellular and multicellular organisms in the same group, the 2 kingdom classification. Example, Chlamydomonas and Spirogyra, they both were placed under algae. And it did not differentiate. So the last setback of this 2 kingdom classification is fungi is heterotrophic and plants are autotrophic. It did not differentiate between this heterotrophic fungi and autotrophic green plants. So in 2 kingdom classification, the heterotrophic fungi and autotrophic green plants were placed under the same kingdom, plant kingdom. But we know at present fungi is made up of chitinous cell wall, while the green plants have cellulosic cell wall. So even though both of them have cell wall, chemically they are different. This is the present information that we have. And this was not available for Linnaeus. So instead what he did was he placed fungi under plants, even though they were heterotrophic and plants were autotrophic, only on the basis of the cell wall he placed. So these are the drawbacks or limitations of two-kingdom classification. So the five-kingdom classification was put forth by R.H. Whittaker in 1969. So there was three-kingdom, four-kingdom classification. We'll not go into those details of it because NCERT has focused on five-kingdom classification. At present, there is also six-kingdom classification where kingdom Monera, now they have split it into kingdom Archaeans or Archaebacteria and kingdom Eubacteria as per six-kingdom classification, which is not included in our syllabus. Anyway, so for information we will be knowing about it. So the five-kingdom classification was put forth by R.H. Whittaker in 1969. So it includes Monera, Protista, fungi, plantae and Animalia. So what were the characters on the basis of which Whittaker classified this five-kingdom classification? The various parameters that he chose was structure of cell, thallus organization, the mode of nutrition, especially bacteria shows maximum mode of nutrition, the type of reproduction, and phylogenetic relationship, that is the evolutionary relationship, was taken into consideration by five-kingdom classification, which was put forth by R.H. Whittaker, Robert Whittaker. So all the living organisms were classified into five kingdoms: kingdom Monera, kingdom Protista, kingdom fungi, kingdom plantae, and kingdom Animalia. Now this table is there in our NCERT book. So on the basis of which the five kingdoms, the different parameters are there. Some of them are unique to them. If you notice here, the cell type is prokaryotic only in kingdom Monera. The remaining four kingdoms are eukaryotic. So four kingdoms are eukaryotic and only one kingdom is prokaryotic. Now the cell wall, if you look into the composition of cell wall, Monera has non-cellulosic. They don't have cellulose. They are made up of polysaccharide and amino acid. See, the table should be read because many information which is not available in the textbook, they are in the text content, they are in the table or in the diagrams. So here, Protista, the cell wall is present in a few of them, not in all of them. And in five kingdom, they are present, but they are having cell wall is present, but it is made up of chitin. In plant kingdom, the cell wall is made up of cellulose. So cell wall is absent in Animalia. Now here, each of this group, so they can take these four attributes and make it as incorrect statement or correct statement questions. Any one of them they can just skip it or make it as a wrong statement and ask you to find the incorrect statement, or they might have given you a correct statement question where three statements are correct and one statement would be so for three statements would be incorrect and one statement would be correct in correct statement questions. So you should be prepared for those sort of questions when you are reading this content. So now after the cell wall, nuclear membrane is absent in Monera, whereas nuclear membrane and nucleolus is present in the remaining four kingdoms. Body organization in Monera, mostly they are unicellular. Protista, a few of them are, so even in Protista, most of them are cellular organization. Fungi, we find it to be multicellular, but it is made up of loose tissue. Plants, they have tissues or organs. And in case of Animalia, there is not only tissues and organs, but there will also be organ system. So the mode of nutrition in Monerans, they have maximum, especially eubacterians, they exhibit maximum type of nutrition mode. So the mode of nutrition is autotrophic. So chemosynthetic and photosynthetic, and heterotrophic will be saprophytic or parasitic. So in our book, they have not differentiated between parasite and pathogen much. So but we have to, even the pathogens, they call it as parasite. But if you look into the difference between parasite and pathogen, it is hairline difference, the parasite and pathogen. So all pathogens cause diseases, and whereas the all parasites need not cause disease. That is the difference between them. So all parasites need not cause disease, whereas all pathogens, they cause disease. That is the difference, major difference between them. So the mode of nutrition in Protista, which includes the Chrysophyta, dinoflagellates, euglenoids, lime molds, and protozoa and protists, most of them, they exhibit autotrophic mode of nutrition, photosynthetic it is, and heterotrophic mode of nutrition. In fungi, it is heterotrophic, either saprophytic or parasitic. In plants, it is autotrophic or photosynthetic. So in case of Animalia, heterotrophic mode of nutrition it is, where none of the organisms are photosynthetic. So mostly their nutrition is holozoic or saprophytic. So these are the attributes on the basis of which the 5 kingdoms are. Each one of them are unique in a few of the other attributes, and they have a clear-cut distinction between them. So as per the parameters put forth by R.H. Whitaker, so these are the major differences between the 5 kingdoms. So the next first kingdom that we are going to study is kingdom Monera or bacteria. In the backdrop, you can see the Bacillus bacteria. So most of them are rod-shaped bacteria, Bacillus bacterium. So we'll be discussing about the kingdom Monerans. Kingdom Monera has all the attributes of prokaryotes. So if you want to remember the major difference between prokaryotes, you should remember that they have an incipient nucleus or false nucleus. That is, they do not have nuclear membrane and nucleolus. Prokaryotes, they do not have cell organelles like Golgi complex, mitochondria, endoplasmic reticulum, lysosomes. They are all absent. And ribosomes are of 70S type. If you remember, 70S type with a larger subunit of 50S and a smaller subunit of 30S. See, these are not normal numerals that we can add them. They are all the sedimentation coefficient values. S stands for Swedberg, the name of the scientist. So these are a few important attributes of prokaryotes. Only one kingdom exhibits prokaryotic nature as per 5 kingdom classification, and that is kingdom Monera. So bacteria are the most abundant microorganisms. So hundreds of bacteria are present in a handful of soil. If we had a microscopic eye, so just like this guy, we would be surprised to see so much of microorganisms in and around us. So they also live in this microorganisms live in extreme habitats such as hot springs, deserts, snow, and deep oceans, especially the archaebacterians. So hot springs, you can find the thermoacidophiles. Then in high salinity, you find the archaebacterians, which we call them as halophiles. And they are also found in marshy habitat as well as in methane, the guts of certain ruminant animals like cows and cattle. You find them, which we call it as methanogens. If you recall, we are using Thermus aquaticus DNA polymerase for PCR process because they have to withstand high temperature. And this Thermus aquaticus is also a type of archaebacterium. So the DNA polymerase of Thermus aquaticus can withstand very high temperatures. That is the reason we are using Taq polymerase in case of the PCR technique, that is the polymerase chain reaction. Anyway, many of the bacteria are parasitic. When we mean parasites, most of them are pathogens. You can see the slides here of variety of bacteria, like there is circular bacteria, there is rod-shaped bacteria, there is spiral-shaped bacteria. So here in the bacteria you can find large number of flagella. In our NCERT book there is no classification of bacteria based on the types of flagella. So types of flagella, there is a classification like monotrichus, lophotrichus, amphitrichus, peritrichus, and atrichus. Maybe in the higher classes we'll be studying about that, but our NCERT syllabus doesn't have information on that. If any bacteria does not have flagella, we call it as atrichous. So we call it as atrichous. You can just remember that. Now, based on the shapes of bacteria, there are bacteria of different shapes. If they are spherical, coccus bacteria. If they are rod-shaped, bacillus bacteria. If they are comma-shaped, Vibrium or Vibrio. So Vibrio cholerae. Cholera is caused by this comma-shaped bacteria. Spirillum, if it is spiral-shaped. So these are the 4 types of bacteria based on their shape. Coccus, it is singular noun. Cocci is plural. Bacillus, bacilli. Vibrium, Vibrio. Spirillum, spirili. So these are the different types of bacteria based on their shape. You have some examples here, like Streptococcus pneumoniae, which causes, it is spherical in shape. You just observe that it causes pneumonia, and we would have studied them under Griffith's experiment. Then Clostridium tetani, which causes tetanus. Then we have this Treponema pallidum. So you might have studied this under the sexually transmitted diseases, especially syphilis is caused by Treponema pallidum. Then we have this Vibrio cholerae, the comma shape, which causes cholera. Streptococcus aureus. These are different varieties. Clostridium botulinum, it causes botulism. It's a food poisoning, especially in canned foods. The Clostridium botulinum is present. It causes botulinism. That is food poisoning it causes. So then there is the other varieties like Salmonella typhi, which causes typhoid. So Helicobacter pylori. So one of the scientists, Barry Marshall of Australia, he swallowed this Helicobacter pylori to just prove the point that Helicobacter pylori causes gastric ulcer. And later he took antibiotics and he was cured from this Helicobacter pylori, that is the gastric ulcer. So he exhibited by this experiment where he conducted an experiment on himself and showed to the world that Helicobacter pylori is majorly responsible for gastric ulcers, and he got Nobel Prize for this work. So you can just remember that. Bacteria structure is very simple. So as I showed you, they are complex in behavior and show metabolic diversity. So some bacteria are autotrophic. They synthesize food from inorganic substrates. The autotrophic can be photosynthetic autotrophs or chemosynthetic autotrophs. Majority of the bacteria are heterotrophs. They do not synthesize the food but are dependent on other organisms or a dead organic matter for food. So the structure of prokaryotic cells we would be again discussing in detail in case of the cell unit of life. But anyway, the glycocalyx is made up of capsule, cell wall and plasma membrane. The cell envelope, the capsule, cell wall and plasma membrane. Then there is a cytoplasm. The circular naked DNA, nucleoid we call it as. Then ribosomes of 70S type are there, plasmids are there. The finger-like protrusions we call it as pili. Pili plays an important role, especially in the parasexual reproduction, conjugation. Unfortunately, we don't have that for our discussion. Conjugation, transformation, transduction. But transformation process, we'll be studying it in 12th as an evidence for DNA to be a genetic material. So then there are also fimbriae which helps in attaching to the substratum. So these are some major features of the bacteria. So these are the diagrams that they have given in your book. So diagram oriented question they might ask you. They might label it as A, B, C, D and ask you to label them. So you should be aware about bacteria of different shapes as per your NCERT book. NCERT book has also given this nostoc where you can find the mucilaginous sheath, heterocyst with 2 polar nodules they are having there. So heterocyst helps in fixing the atmospheric nitrogen. So a filamentous blue-green algae, nostoc. See, nostoc has a nickname. It is also called as moon spit. So very few of them know that it has a nickname, moon spit. So this is the dividing bacteria which is showing fission. So binary fission, where the nucleus directly divides into two lobes and moves towards the opposite pole, and there is a formation of two cells. So this we call it as the binary fission. Now the next one is the classification of bacteria. So bacteria are majorly classified into two types: archaebacterians, halophiles. These are archaebacterians which are found growing in high saline condition. Thermoacidophiles are archaebacterians found growing in very high temperatures, and archaebacterians found growing in marshy region or in the guts of ruminants are methanogens. So eubacteria, based on their mode of nutrition, they are of two major types: autotrophs, heterotrophs. Autotrophs, if they utilize sunlight as a source of energy, we call it as photosynthetic autotrophs. The chemosynthetic autotrophs, they require energy liberated during the oxidation process, and that is how they synthesize food. So we call them as chemosynthetic autotrophs. Then there are heterotrophs, the parasitic heterotrophs and saprophytic heterotrophs are there. So here in this slide you can see that if they are living in harsh and extreme habitats, we call them as halophiles. That is hot springs in extreme salty areas, halophiles. Hot springs, we call them as thermoacidophiles. And if they are seen in marshy area, we call them as methanogens, or in the guts of ruminants also they are seen. So archaebacteria have a different cell wall structure for their survival in extreme conditions. That is the reason in 6 kingdom classification, archaebacterians, they are given a separate kingdom itself. So they are promoted as kingdom archaeans. So since they are different cell wall structure for their survival in extreme conditions, methanogens are present in the guts of ruminant animals like cattle, the cows, buffaloes, etc. They produce methane biogas from the dung of these animals. So eubacteria, these are true bacteria having a rigid cell wall and a flagellum. So you can notice in some of them they are having a rigid cell wall and a flagellum if motile. They include autotrophs, that is photosynthetic autotrophs, as well as chemosynthetic autotrophs, and they also have heterotrophs. So photosynthetic autotrophs, example is cyanobacteria. So cyanobacteria, or earlier they were called as cyanophyceae or blue-green algae or mixophyceae. These were the earlier names. Presently we call them as cyanobacteria. Earlier they were called as cyanophyceae or mixophyceae, commonly called as blue-green algae, but at present they are called as cyanobacteria. So you can notice that they also have chlorophyll A similar to that of green plants. But why are they placed separately, and why they are not placed under this sort of question can be asked in NCR, NEET exams. Why are the blue-green algae not placed along with chlorophyceae, phaeophyceae and rhodophyceae in plant kingdom? So the major reason for that is blue-green algae is the prokaryotic organism, whereas all those three, the chlorophyceae, phaeophyceae and rhodophyceae, they are eukaryotic organisms with cellulosic cell wall, and they have chlorophyll A and other pigments. That is the reason we have placed them under plant kingdom, whereas blue-green algae or cyanobacteria is placed under kingdom Monera because of its prokaryotic nature. Chlorophyceae, phaeophyceae, rhodophyceae are the eukaryotic nature they have. So that is one point that you should remember about, and so they might ask you during the NEET examination. Cyanobacteria, blue-green algae, they can be unicellular like Chlamydomonas, colonial like volvox, or they can be filamentous, marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. So cyanobacteria, unicellular, colonial or filamentous. Here they have nostoc oscillatoria. So the colonial form volvox, what I said, is in green algae. Filamentous also, I gave an example of green algae, but in blue-green algae, the filamentous form, what you can remember is the oscillatoria. Nostoc is also filamentous form. Unicellular, we can remember the Anabina. Some of them are unicellular varieties. So the colonies are generally surrounded by gelatinous sheath. They often form water blooms. So if water is polluted and they look bluish green in color, and you take a sample of that water and observe under microscope, you find large varieties of this cyanobacteria present there. So polluted water, we call it as water blooms, and especially if it is caused by cyanobacteria, we call it as cyanophycean blooms. Some of them fix atmospheric, that is some of the blue-green algae fix atmospheric nitrogen in specialized cells called heterosis. In the diagram I had marked that heterosis with two polar nodules. So examples are Nostoc and Anabina. But if you remember, in coralloid roots of gymnosperms, they are some of the roots which are near to the surface of the soil. These coralloid roots have a special function. They can fix the atmospheric nitrogen. Why do you think they can fix the atmospheric nitrogen? Because they have a Anabina cycadae, the blue-green algae symbiotic association in the coralloid roots of gymnosperm. Anabina cycadae, we call it as, which is responsible for the atmospheric nitrogen to be fixed in the coralloid roots of gymnosperm plant cycads. So chemosynthetic autotrophs, they oxidize inorganic substances such as nitrates, nitrites and ammonia and use the energy released for synthesis of ATP. So whereas in photosynthetic autotrophs, they use the energy from the sunlight for synthesis of ATP. But here the energy is obtained from oxidation of inorganic substances such as nitrate, nitrite and ammonia. So they also, the chemosynthetic bacteria help in recycling nutrients like nitrogen, phosphorus, iron and sulfur. So these are the roles of them. So heterotrophic bacteria, they are the most abundant type of bacteria in nature. The majority of these heterotrophic bacteria are decomposers. So they can frame some incorrect statement using these points of heterotrophic bacteria. So the impact of bacteria on human affairs is they are used to make curds from milk, production of antibiotics. So many of the antibiotics are obtained from the bacteria, fixing nitrogen in leguminous roots, especially rhizobium bacteria. So fixing nitrogen in leguminous root by rhizobium bacteria is also one of the important functions which benefits human beings. Many of them are pathogens causing diseases like cholera, typhoid, tetanus, citrus canker. Citrus canker is caused by Xanthomonas citri. So the Xanthomonas citri causes the citrus canker. So this is seen in plants. Then one of the host, Agrobacterium tumefaciens, which we use in our biotechnology. Agrobacterium tumefaciens causes crown gall disease. They cause tumors in all these dicot plants. Crown gall disease is caused by this bacteria, Agrobacterium tumefaciens. So reproduction in bacteria is mainly by fission. So reproduction in bacteria is mainly by fission. Under favorable conditions, they also produce spores. Unfavorable conditions, in order to overcome the unfavorable conditions, they develop true walls and form endospores, most of the bacteria. So the bacteria also reproduce by a primitive type of sexual reproduction, which involves only DNA transfer. There is no formation of gametes or gametogenesis, no such process at all. Directly there is a transfer of DNA. And you should remember under this there are 3 steps like conjugation, Transformation. Transformation you have studied in DNA for an evidence to be as genetic material. Then transduction. In transduction, the DNA from one bacteria is transferred to another through the bacteriophage, a virus. So that process we call it as transduction. The ultimate purpose of sexual reproduction is not mating, intercourse, or the formation of gametes or fusion of gametes. The ultimate purpose of sexual reproduction is DNA should be transferred. There should be a transfer of DNA and recombination of genes. That is the ultimate purpose, so that it leads to variation. And you know that variations, apart from mutations, are raw materials for evolution. So this point you just have to remember, where bacteria exhibits primitive type of sexual reproduction. Mycoplasma, at present you might have heard about new pneumonia that is spreading very rapidly in China, and it is almost like a pandemic status in China. That is caused by mycoplasma. See, mycoplasma is now creating a havoc in China by infecting the children with pneumonia-like symptoms. So mycoplasmas, they are also prokaryotic organisms. They are organisms without a cell wall. So if you look into the diagram, you can see that they are organisms without cell wall. So they are the smallest living cells known. They can survive without oxygen. Many are pathogenic in animals and plants. So once they had asked, if you look into the DNA here, it is not circular, it is linear. So linear type of DNA is seen in dash prokaryotes. So mycoplasma, you can just remember about. So kingdom Protista, the second kingdom that we will be discussing is kingdom Protista. It includes single-celled eukaryotes. So unicellular eukaryotes. So for the first time you find eukaryotic organisms. So they have all the eukaryotic organism features, like they have nucleus and nucleolus. They have double membrane cell organelles like mitochondria, Golgi complex, endoplasmic reticulum, lysosomes, chloroplast. They are all present. Some of these protists have flagella or cilia, and protista also have ribosomes of ATS type. So ribosomes of ATS type with a larger subunit of 60S and a smaller subunit of 40S. So they have ATS type of ribosomes with a larger subunit of 60S and smaller subunit of 40S. And also what you have to remember is the ribosomes in case of mitochondria and chloroplast will be of 70S type. And you also should understand that mitochondria and chloroplast also has DNA. So they can live outside of the cell as a separate entities. So if they ask you cell organelles which can live as a separate entities, you can always write chloroplast and mitochondria, because they have their own ribosomes and they also have their own DNA also. So but ribosomes within this eukaryotic cell, the cell organelle like mitochondria and chloroplast, they will be having 70S type. Only in the cytoplasm you find the 80S type of ribosomes in eukaryotes. So that point you should be very clear about. So protists are primarily aquatic. So they are found mostly in water. It is a link with plants, animals and fungi. Almost a connecting link between plants, animals and fungi is protista. They reproduce asexually and sexually. So there is cell fusion and zygote formation. Now classification of this protista, if you look, chrysophytes. So chrysophytes includes diatoms and golden algae. Dinoflagellates, you can see this colorful variety of dinoflagellates like red dinoflagellate, blue dinoflagellate, pink dinoflagellate, green dinoflagellate, yellow dinoflagellate. So dinoflagellates, especially goniolates, you should remember about, which causes red tide. And we have euglenoids. Euglenoids, they are having flagella, two flagella, unequal flagella. One is short another is long flagellum. So they do not have cell wall. They have a flexible proteinaceous layer called as pellicle. So the euglenoids, they also exhibit mixotrophic nutrition. This point is not given in NCERT, but you should be aware about this. Mixotrophic nutrition implies in presence of sunlight, euglenoids undergo photosynthesis. In absence of sunlight, they are predators. So this is what we mean by mixotrophic nutrition. Slime molds, these are slime molds. So they are found growing on the decaying matters. So they are saprophytes. Then protozoans. Under protozoans we are going to study based on their locomotor structures. We have 4 types. That is the amoeboid protozoans, example amoeba and entamoeba. Then we have the flagellate protozoans like the trypanosoma gambiensis, which causes sleeping sickness, and the vector which spreads this CC fly. You should remember about that. Then there is also Leishmania donovani, which causes kala azar or black fever. Even they are a protozoan having flagella. Then under ciliate protozoans, we are going to study about paramecium. So here you can see that paramecium diagram, which is also commonly called as slipper animalcule. And the last one is the sporozoans. Sporozoans do not have any locomotor structure. So here you find Plasmodium vivax, Plasmodium malariae, Plasmodium falciparum. So they are all the different types of sporozoans, non-motile sporozoans. And these are the pathogens which cause malaria. Mosquitoes don't cause malaria. They are just a vector which spreads these Plasmodium or the pathogens which causes the malaria from an infected person to healthy person. They are just a vector, and especially female mosquitoes are blood sucking, and they are the ones which spread the diseases. So chrysophytes, they are found in freshwater and marine environments. So many times they are also called as jewels of the plant kingdom, especially diatoms, jewels of the plant kingdom, because of the ornamentation that they have, the design pattern that they have. They are called as jewels of the plant kingdom. They are found in freshwater and marine environments. Chrysophytes are microscopic and they float passively in water currents. So planktons are floating microscopic organism. So most of the chrysophytes are photosynthetic. It includes diatoms and golden algae. So you can see this design pattern of the diatoms, which are called as jewels of the plant kingdom. So the golden algae, you can see them. So these are a few examples of the chrysophytes. So chrysophytes, the diatomaceous earth is formed, which are nothing but the fossilized forms of the diatoms from lakhs or millions of years ago. Since their cell wall is made up of silica, they can withstand, even though the organic contents are degraded, the cell wall are retained as it is. A soil which is rich in this sort of diatoms, we call it as diatomaceous earth, or Kaisel Gur, they also call it as. So they have siliceous cell walls forming 2 thin overlapping shells. One we call it as epitheca, the other one is hypotheca. They fit into each other just like a soapbox. So the larger one is epitheca, the smaller one is hypotheca. At degree level we discussed about this. Then they would also be having ornamentation. And I told you this design pattern itself makes them to be called as jewels of the plant kingdom. You can see the design pattern here. So such wonderful design patterns they have. So diatoms are the chief producers in the oceans. Whenever we are talking about planktons in oceans, as a producer, you should remember the example of diatoms. So diatoms are the chief producers in the oceans. So diatoms have chief producers in the ocean. The cell wall deposit of diatoms over billions of years in the habitat is known as diatomaceous earth. You can see the powder, diatomaceous powder here. So this is diatomaceous earth. This diatomaceous earth is used in polishing. It is used in filtration of oil, and in manufacture of syrups they use this diatomaceous earth. If you observe it under electron microscope, the diatomaceous earth, you can see the siliceous cell wall. Since the siliceous cell wall is not degraded, they are the past remnants of diatoms. So billions of years ago they might have existed. All their organic contents are lost except for the siliceous cell wall, which is found in this diatomaceous earth. Dinoflagellates, they are very colorful. They look almost like tap-like structures. So they are very colorful. So mostly marine and photosynthetic dinoflagellates. They appear yellow, green, brown, blue or red depending on the pigments in the cells. The cell wall has stiff cellulose plates. So you can see this stiff cellulose plates together. They have two types of flagellum, a transverse flagellum and a longitudinal flagellum. So they also, the Goniolax, I told you, it causes red tide. Okay, the red tide, it kills all the aquatic organisms, flora and fauna within that red tide because it secretes toxic substances. Most of them, dinoflagellates, they have two flagella. So you can see one lies horizontally, one is longitudinally arranged and the other is transversely arranged in a furrow. So you can see this transverse flagellum and this is the longitudinal flagellum. Red dinoflagellates, example, you should remember Goniolax, which is a red dinoflagellate, undergoes rapid multiplication in the sea. You can see the red tide here, so that the sea appears red and they release toxic substances which kill the marine animals like fishes. So euglenoids, they are mainly freshwater organisms. They are found in stagnant water which is not moving. You can find this euglenoids. They have a long flagellum and a very short flagellum which is usually not visible. Instead of cell wall, the euglenoids have a protein-rich layer called pellicle. So pellicle gives flexibility to the body. So now they can frame assertion and reason question here. So euglenoids have a flexible body. Reason, instead of cell wall, they are made up of a protein-rich layer called pellicle that is responsible for flexibility. Both A and R are correct, and R is the correct explanation for E. So they can make that. They have two flagella, a short flagella and a long flagella. So euglenoids are photosynthetic in the presence of sunlight. In the absence of sunlight, they behave like heterotrophs by predating on other organisms. So they might give you assertion and reason here. Euglenoids exhibit mixotrophic nutrition. So mixotrophic nutrition. Reason, in presence of sunlight, they undergo photosynthesis, and in absence of sunlight, they are heterotrophs which predate on other organisms. So if reason is given in such a way, then you can answer it as both A and R are correct, and R is the correct explanation for A. Example is euglena. Slide modes are quite interesting. They have not given any example. This is the diagram that they have given in our textbook, NCERT book. So, but in reality, this is how the slime molds look, almost like yippee noodles for you. It looks like, and you find that they spread to large areas. So this aggregate of slime molds, we call it as plasmodium. Don't get confused between the plasmodium of the protozoan protist, that is the malarial organism pathogen. If we are talking about that plasmodium, P will be capital and it will be in italics. So whereas if you are talking about normal word aggregate of slime molds, plasmodium is P is small letter and it is normal script. It is not a scientific name. So that is how you should be able to differentiate between the generic plasmodium and normal word plasmodium. So they are saprophytic protist slime molds. They are dependent upon dead and decaying remnants of plants. The body moves along decaying twigs and leaves, engulfing the organic material. So under suitable conditions, they form an aggregation. What do we call this aggregation as? Plasmodium. And it may spread over several feet. So under unfavorable condition, plasmodium differentiates and they form fruiting bodies. So they bear the spores at their tips. So they are forming the fruiting bodies which have spores at their tip. So the spores have true walls so that they can withstand extreme conditions. And the true walls of the spores are highly resistant, and these spores can survive for many years. So that's how the slime molds survive and exist for many years, and they are going to become functional at the onset of favorable conditions. Spores are also carried by air current. So now the final part of the discussion for the protista is protozoans. So we have amoeboid protozoans, flagellated protozoans, ciliated protozoans, and sporozoans. So amoeboid protozoans, they have locomotory structure of false feet, which we call it as pseudopodia. So flagellated protozoans, they have the locomotory structure in the form of flagellum. The example that they have given for us is trypanosoma, which causes sleeping sickness, and it is spread from an infected person to healthy person by a vector, which we call it as CC vector. Then ciliated protozoans, we have the paramecium, and sporozoans, we have the plasmodium. So amoeboid protozoans, now these sentences, they can be framed as incorrect or correct statements. So we should be aware about all the statements. Live in fresh water, sea water or moist soil. They move and capture prey by putting out pseudopodia. Pseudopodia is not only helpful in locomotion but also in forming food vacuole. They help in capturing the prey. Example, amoeba, Proteus. Marine forms of the amoeboid protozoans, they have silica shells on their surface to prevent the salinity. So some of the amoeboid protozoans are parasitic, especially Entamoeba histolytica. Entamoeba causes amoebiosis. Then flagellated protozoans, they are free living or parasitic. They have flagella. Parasite forms causes disease like sleeping sickness. Example, trypanosoma. And I had also told you that the vector which spreads this disease from infected to healthy person is CC fly. You can see this CC fly here. The ciliated protozoans, they are aquatic, actively moving cells using cilia. They have a cavity, which we call it as gullet. So you can see this cavity gullet. So cilia not only helps in locomotion, but it also guides it through the cell mouth or cytopharynx. The food is guided into the cell mouth or cytopharynx by the cilia. So due to the movement of cilia, water with food enters into the gullet. Example, paramecium, which is commonly called as slipper animalcule. And if you look into the practicals, you would have drawn the diagrams, macronucleus, which is kidney shaped, and micronucleus, which is small spherical nucleus. So they are binucleate. So all this you are aware about, and you would have drawn it in your practical records. So sporozoans, they have an infectious spore-like stage in their life cycle. Example is plasmodium, which causes malaria. So there is Plasmodium falciparum, Plasmodium vivum, Plasmodium malarium, Plasmodium ovale. These are all just for your information, you can be aware about it. So sporozoans, they are non-motile. They have an infectious spore-like stage in their life cycle. Example is Plasmodium, so which causes malaria. So mosquito, it just spreads the disease from an infected person to healthy person. That is why we call it as vector. So these are the diagrams for dinoflagellates in our NCERT book. The paramecium diagram, slime molds, then the paramecium diagram of it. So euglena it is here, not paramecium, slime mold, and this is paramecium. So kingdom fungi. So you can see at the backdrop, very colorful fungi. The more colorful and attractive they are, avoid them when talking about the fungi. So you should understand that fungi are having a cell wall, but it is made up of chitin. They have reserve food material, but it won't be starch, it would be in the form of glycogen. So they have a cell wall but made up of chitin. and they are heterotrophic in function. They are achlorophyllous. They do not have chloroplasts. They cannot synthesize their own food. They are either dependent upon dead or decaying organism as a saprophyte, or they are parasitic, or they cause diseases. So many plant diseases are also caused by fungi. So we'll also discuss about all those in this particular slide. The unique kingdom of heterotrophic organism. So they are cosmopolitan in distribution. So almost everywhere you find them this. So they are growing in warm and humid places. So example for the fungi are bread mold, orange rots, mushroom, toadstools. So you can see here the bread mold, which is black in color, and you have this orange rots. The juicy endocarp becomes very dry if they are infected by this orange rots, which is caused by fungi. The edible mushroom, we call it as Agaricus bisporus. So the toadstool. So toadstool is also highly poisonous, even though it looks very colorful and attractive. So the white spots on mustard leaf is caused by a parasitic fungus. See, some of the fungi are also source of antibiotics. Penicillium notatum, the wonder drug, especially during the Second World War, it saved a lot of lives. So many of the soldiers used to die because of poisoning of wounds. But this Penicillium notatum, from which the penicillin was extracted, it saved large number of lives. So that is why it is called as wonder drug. It was discovered accidentally by Sir Alexander Fleming. You know about that. So Penicillium roqueforti. So probably for the Roquefort cheese, there is also one more species that you would have studied in the zoology part of it. See, some unicellular fungi like yeast are used to make bread and beer. So unicellular fungi, Saccharomyces, we generally have a generic name, Saccharomyces. Other fungi cause diseases in plants and animals, especially wheat rust causing puccinia. So you can see the rust disease here on the leaf. So these are the conidia formation in Penicillium. You can see the budding stages in yeast. So yeast multiplies by budding, so which is amitotic or direct cell division, we call it as. Except yeast, fungi are filamentous. Their bodies consist of long, slender, thread-like structures, which we call it as hyphae, and a network of hyphae or collection of hyphae, we call it as mycelium. The branch of biology which deals with the study of fungi, we call it as mycology. The word mycology is derived from mycelium. So the network of hyphae is known as mycelium. Hyphae are of two types: cenocytic hyphae, so where you find that there are no cross walls in between them and they are all free. You find the multinucleate cytoplasm. So they are continuous, cenocytic hyphae, they are continuous tubes filled with multinucleated cytoplasm. Or you can also call it as aseptate hyphae, cenocytic or aseptate hyphae. In septate hyphae, they have septa or cross walls. So the fungal cell wall, unique thing, this you should remember, it is made up of chitin and polysaccharides. Even though they have a cell wall, they are made up of chitin and polysaccharides. So mostly fungi are saprophytic. So now you can see that they are found growing on the log of woods. So decaying logs of wood, you find the fruiting bodies, isn't it? So they absorb soluble organic matter from dead substrate. Some of them are parasites, they can cause disease. Some of them live as symbionts, like an example is lichens or lichens, however you pronounce it as. Fungi plus algae. So fungal component is mycobiont. The algal component is phycobiont. So there is a symbiotic association between them, wherein algae synthesizes carbohydrates and the fungi provides them with water and nutrients. So both of them are getting benefited from each other. So lichens are a very good example for mutualism or symbiotic association. Even mycorrhiza, they are fungi which are found associated with roots of higher plants. and they also help in providing for the host plant the water and minerals, and in turn they get the food from the roots of these plants. So there is a symbiotic association that we find between fungi and other organisms. If it is fungi and algae, lichens; fungi with roots of higher plants, so mycorrhiza. The reproduction types in fungi is of different types. Vegetative reproduction is by fragmentation. If hyphae breaks into fragments, they form new organism, new hyphae is formed, that is new fungi is formed. Fission is binary fission and budding. It is a direct cell division where the nucleus enlarges in size and it breaks into two lobes, and one of the lobes moves into the bud in budding, or it moves towards the opposite poles and they form two cells. There are no cell division stages like prophase, metaphase, anaphase and telophase. So fission and budding is, that is the reason they are called as direct cell division or amitotic cell division. So in this we notice that. So asexual reproduction by asexual spores, conidia formation. Conidia formation is asexual spores which are formed on special stalks called as conidiophores. They are exogenously cut off, the conidia, and when this conidia falls onto a new place, they germinate to form a new hyphae or fungi. Sporangiospores, they are spore-like structures produced in sporangium. Zoospores, they are motile, usually biflagellate. They are produced within zoosporangium, and each zoospore can germinate to produce a new fungi. So since there is no formation of gametes or fertilization, this is an asexual method of reproduction. Conidia formation, sporangiospores and zoospores are asexual method of reproduction. Sexual reproduction by sexual spores, especially oospores, ascospores, basidiospores. I'll be telling you how the difference between ascospores and basidiospores. We'll be discussing about that. And sexual reproduction in fungi involves three steps. Fusion of cytoplasm, plasmogamy. Then they might exist in a resting stage, that is N+N condition or dikaryotic phase. Then plasmogamy is followed by karyogamy, fusion of nucleus, and immediately after the fusion of nucleus, they immediately break down to form haploid spores. So that is what they become haploid by undergoing meiosis. So there is plasmogamy, karyogamy and meiosis of zygote. So all these are happening in case of the special type of sexual reproduction in fungi. So you can see this slides for vegetative propagation. This is by breaking up of the hyphae, each hyphae forms a new fungi. So you can see fission in yeast, Saccharomyces. The nucleus enlarges in size, it becomes dumbbell shaped. They break into two lobes and moves towards the opposite poles, and there are two daughter cells formed. And here there is no prophase, metaphase, anaphase, telophase stages. Directly the nucleus and cytoplasm divides. That is why fission, budding, they are all examples for direct cell division. So in budding in yeast, even here the nucleus which is enlarged breaks into two lobes. One of the lobes moves towards a small protrusion called as bud, and then the cross wall is formed. This bud, if it gets detached, they form a new yeast cell. Or many times this bud again gets budding and there will be again budding, and they form a false mycelium or pseudo-hyphae like structure are formed. So when the buds don't get detached from the parent cell, it looks like a false hyphae or mycelium. So we call it a pseudo-hyphae or pseudo-mycelium if the buds don't get detached in yeast. So asexual reproduction is by spores such as conidia, sporangiospores and zoospores. So sexual reproduction is by the formation of oospores. Oosporangium forms oospores, then asci or ascus, they produce ascospores. Basidium produces basidiospores. They are produced in distinct fruiting bodies. So now ascospores are produced in ascocarp. The fruiting body is ascocarp. Basidiospores are produced in basidiocarp. So they are produced in distinct fruiting bodies. Sexual reproduction, there are three steps in sexual reproduction. plasmogamy, karyogamy, and meiosis of zygote resulting in haploid spores. Plasmogamy is fusion of protoplasm of two motile or non-motile gametes. Karyogamy is in many of these basidiomycetes and ascomycetes, the nucleus immediately after plasmogamy they do not fuse. They exist in a dikaryotic phase or dikaryon phase. Then later they undergo fusion. So that is what we find in case of ascomycetes and basidiomycetes. The zygote that is formed immediately undergoes meiosis, resulting in haploid spores. So this is a unique type of sexual reproduction which you find in case of ascomycetes, basidiomycetes, especially the fruiting bodies ascocarp and basidiocarp. In sexual reproduction, two hyphae, haploid hyphae, come together and fuse. In some fungi, fusion of two haploid cells immediately results in diploid cells. In ascomycetes and basidiomycetes, a dikaryotic stage or dikaryophase, that is N plus N, two nuclei per cell occurs. So we call such a condition as dikaryon. So later parental nuclei fuse and the cell becomes diploid. Fungi forms fruiting bodies in which reduction division occurs, leading to formation of haploid spores, especially in ascocarp. They produce ascus. Ascus produces 8 ascospores endogenously, that is within. Now in case of the fruiting body basidiocarp, they produce basidium. In basidium, 4 basidiospores are produced exogenously. They are produced. Externally they are cut off. Just like conidia is also exogenously produced. So this is the difference between ascomycetes and the basidiomycetes type of formation of spores. So 8 ascospores produced endogenously in case of ascomycetes, whereas 4 basidiospores produced exogenously in case of basidiomycetes. So based on morphology of mycelium, mode of spore formation and fruiting bodies, fungi are classified as, I ask my students to remember it as PABD, P-A-B-D. Phycomycetes, lower fungi. Phycomycetes is called as lower fungi. Ascomycetes, cup or sac fungi, they call it as. Basidiomycetes, some books, they refer it as club fungi. Deuteromycetes, imperfect fungi. Imperfect fungi because they do not exhibit any sexual reproduction stages in Deuteromycetes. So we have here the bread mold, then there is yeast, the agaricus, then you can find the coletotrichum, alternaria, and the trichoderma in case of Deuteromycetes or imperfect fungi. So let us discuss each one of them. So Phycomycetes or the lower fungi. So Phycomycetes is the only only fungi which has cenocytic fungi, whereas all of them are having the septate fungi. So that we should be aware about. See, Phycomycetes has cenocytic or aseptate fungi. If you look into Ascomycetes, they have the branched and septate. Basidiomycetes also will be having branched and septate fungi. So, and even the Deuteromycetes will be having branched and septate fungi. So all of them are having it except the Phycomycetes. Phycomycetes is the only fungi which is having cenocytic or aseptate fungi. So this point you should remember. Then conidia formation is seen only in Ascomycetes and Deuteromycetes. So nowhere in the content they would have given such things. So these are implied things which we have to know about. So Phycomycetes, the lower fungi, they occur in aquatic habitats, decaying wood in moist, and they are obligate parasites on plants. The mycelium is aseptate and cenocytic. Mucar example, rhizopus which causes bread mold, then albugo which causes the white spots on mustard. It's a parasitic fungi on mustard. So phycomycetes is asexual reproduction by motile zoospores. Non-motile we call it as aplanospores, and they are produced within sporangium. Zoospores are produced within zoosporangium. Aplanospores are produced in aplanosporangium. So sexual reproduction is fusion of two gametes and they form zygospores. Gametes can be isogamous, similar type, or anisogamous, or oogamous type of gametes you can notice. Ascomycetes, the sac fungi or cup fungi, example for these are the penicillium, yeast, neurospora. So all this comes under the ascomycetes or sac fungi. Unicellular example is Saccharomyces. The multicellular example is penicillium. So mycelium is branched and septate. I told you only one class is unbranched and cenocytic, that is Phycomycetes. So the Ascomycetes are saprophytic, decomposers, parasitic or coprophilous. They are found growing on dungs. So that is why we call it as coprophilous. So Ascomycetes undergo asexual reproduction by conidia formation. Conidia are produced exogenously, that is externally, on a special mycelium called conidiophores. So you can see this conidiophores, they are producing a lot of conidia. These conidia, when they fall on suitable surfaces and under favorable condition, they form new hyphae. Conidia germinate to produce mycelium. So conidia formation is seen in only two classes of fungi. One is Ascomycetes, another is Deuteromycetes. So sexual reproduction in Ascomycetes is by ascospores produced endogenously in a sac called as asci. Asci are arranged to form ascocarps. So, and I have already told you they are produced endogenously. So 8 ascospores are produced endogenously in asci. So the asci are found arranged within ascocarps. Example for Ascomycetes is Aspergillus, Claviceps purpurea, Neurospora. Neurospora is considered to be Drosophila of plant kingdom or Cinderella of plant genetics, because many biochemical and genetic works are done on this Neurospora. So you should remember about Neurospora is called as Drosophila of plant genetics because many biochemical and genetic experiments are worked on it. So they can give it as assertion and reason. So you should be prepared for that. So the edible Ascomycetes, they might ask you a question, especially morels and truffles. So you can see here the morels, which almost looks like a dried date palm, isn't it? But these are delicacies and they are very delicious and they are consumed by the Americans and Europeans. Morels and truffles are edibles and are delicacies. We rarely consume them. But if they ask you, what are the edible Ascomycetes? So you should remember the edible Ascomycetes are morels and truffles. Basidiomycetes, some of them are all very colorful. The more colorful they are, you have to avoid them. I'm talking about again mushroom. So because they are going to lead to many toxic substances are there, hallucinogenic substances are there, so which might be harmful for us. So that is the reason, without any knowledge, we shouldn't consume any mushroom. So time-tested mushrooms should be consumed. So the Basidiomycetes includes mushrooms, bracket fungi, or puffballs. See, such colorful bracket fungi are also there. They look like C-shaped bracket fungi. This is puffball. So these are all the examples for Basidiomycetes, mushroom, bracket fungi, or puffballs. They grow in soil, on logs and trees and stumps. And in living plant bodies, some of the Basidiomycetes, they also cause diseases in plants, like rust in plants and smuts. Rust is caused by Puccinia, smut is caused by Ustilago. They have asked questions regarding this also. Rust is caused by Puccinia, smuts are caused by Ustilago. So this also you should be remembering for your NEET examination. Mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is seen in Basidiomycetes. Sex organs are absent, but plasmogamy occurs by fusion of two vegetative or somatic cells. The resultant structure is dikaryotic. It gives rise to basidium. Karyogamy and meiosis occurs in basidium, producing 4 basidiospores exogenously. In ascospores, there are 8 ascospores produced endogenously, but here in Basidiomycetes, 4 basidiospores are produced externally or exogenously. Basidia are arranged in fruiting body called as basidiocarps. See, what we consume in Agaricus bisporus or button mushroom or milky mushroom is nothing but the fruiting body. The plant body, it is microscopic, which we call it as hyphae. So whatever we are consuming is the fruiting body. So basidia are arranged in fruiting body called basidiocarps. Example, Agaricus bisporus button mushroom. Agaricus is mushroom. Bustilago causes smut. Vaccinia causes rust fungus. So they can give those as match the following column 1 and column 2. Name of the organism and common names. So you should be prepared for that. The last class for fungi is Deuteromycetes or fungi imperfecti, or you can also call it as imperfect fungi. Why do we call that as imperfect fungi is they exhibit asexual reproduction, vegetative reproduction, everything is there, but they do not show any sexual reproduction, or we might not have yet discovered those processes. All the fungi which do not exhibit sexual reproduction, they are placed under this imperfect fungi or Deuteromycetes. So for you, they have given 3 examples to remember for Deuteromycetes. I ask my students to remember by the acronym CAT, Coletotrichum, Alternaria, Trichoderma. So these are the 3 examples that you can remember for imperfect fungi. There is also Sarcospora arachidicula, which causes Tika disease in groundnut, but they have not given it. Earlier in state syllabus, we used to discuss that. So the Deuteromycetes is commonly known as imperfect fungi, and their asexual or vegetative features are known. Perfect stage or sexual stage is not known, or they are not yet discovered. So when perfect stages are discovered, so these members within the Deuteromycetes, they are often moved to Ascomycetes or Basidiomycetes as per their structures. It is also possible that asexual and vegetative stage have been given same place under Deuteromycetes and sexual stages under Phycomycetes, Basidiomycetes, Ascomycetes. That could also be the possibility. So all the asexual and vegetative stage have been given one name and placed under imperfect fungi or Deuteromycetes. Sexual stages, we have categorized them as Phycomycetes, Ascomycetes, Basidiomycetes. When the linkages are established, the fungi are correctly identified and removed out of Deuteromycetes, and we place them either in Basidiomycetes or Ascomycetes. So Deuteromycetes reproduce only by asexual spores, conidia. Mycelium is septate and branched. So some are saprophytic or parasitic. Majority of them are decomposers of litter. Litter is the dried leaves that fall on the ground. We call them as litter. And they help in mineral cycling also. Deuteromycetes helps in mineral cycling. This is unique that we have not seen in other members of fungi. So I ask you to remember all these 3 examples as CAT, Coletotrichum, Alternaria, Trichoderma. So these are the examples for the fungi imperfecti. So these are diagrams that they have given in our textbook. So especially Mucar, then there is Aspergillus, Agaricus bisporus they have given. So the next 2 kingdoms that we are going to discuss about is kingdom Plantae and kingdom Animalia. So what here you can see is both this Plantae and Animalia are placed together. So our dependence on each other is very much tightly linked. So that is why the backdrop of this slide, you can see a panda feeding on the leaves of bamboo. Okay. The next kingdom that we are discussing will be the plants. Without plants or producers, we are nothing at all. So let us proceed with the characters of plants. So they are eukaryotic organism, chlorophyll containing organism with cellulosic cell wall. and they are always autotrophs. Some of them are partial heterotrophs, such as the insectivorous plants, which capture insects through the various leaf modifications, so that they can fulfill the nitrogen deficiency that they are having in the soil where they are growing. Example, bladderwort and Venus flytrap. The main reason for them to trapping the insect is to fulfill their nitrogen requirement, and the soils where they are growing, they don't have nitrogen. So that is why they are partial heterotrophs, but they do undergo photosynthesis also. Parasites such as cuscuta, if you look here, it looks like ippy noodles so they are found completely growing on the host plant. They draw nourishment from the host plant. They don't have leaves. If you look into cuscuta, the roots of it enters into the host plant, vascular tissue especially. They have these button-like structures called as hostoria, through which they absorb the nutrients from the host plant. They don't synthesize their own food, so they are a total parasite. So cuscuta is a total parasite. So they are also called as dodderwort. Common name is dodderwort. So you just remember about them. So they are exceptions to the definition of plants. Usually plants are eukaryotic, autotrophic, chlorophyll-containing organisms with cellulosic cell wall. So some exceptions are like insectivorous plants, which is partially heterotrophic, and parasites like cuscuta, which is completely parasitic, and they are dependent upon the host plant for their nutrients. So under plant kingdom, we will be studying more under this. The plantae includes algae. So here you find at the backdrop Volvox diagram. You can see the bryophytes with the sporophyte foot setae capsule. Then you have the ferns, the gymnosperms, and the angiosperms. So plantae includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Algae, we are going to study three of them: chlorophyceae, then we are going to study phaeophyceae or brown algae, rhodophyceae or red algae under the algae. So they are eukaryotic algae, that is why they are placed under plantae. So life cycle of plant has two phases: diploid sporophytic and haploid gametophytic. So the sporophyte definition is sporophyte. Plants which are producing spores, we call them as sporophytic plants. Plants which are producing gametes, we call it as gametophytic plant. So there is a diploid sporophytic and haploid gametophytic stages in plants. So we find that they alternate with each other, and we call this as alternation of generation. There is an alternation of generation between diploid sporophyte and haploid gametophyte in plants. We'll be studying about this haplodiplontic, diplontic, and the diplontic life cycles. So haplodiplontic, the example for that is bryophytes and pteridophytes. Diplontic example is we have the gymnosperms and angiosperms. Haplontic examples, we have some of the algae. They are all placed under the haplontic life cycle. So that we'll be studying later. So kingdom Animalia, they do not have cell wall. They are heterotrophic organisms, multicellular. See, fungi and algae, the fungi and Animalia, how they can be differentiated is both of them are eukaryotic, both of them are heterotrophic, both of them are multicellular. But fungi has cell wall, Animalia does not have cell wall. That is unique for animal kingdom. So the animal kingdom has wide diversity. Animals are multicellular, heterotrophic, eukaryotic organisms without cell wall. So they directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food as glycogen or fat. In plants, they store food as starch. The mode of nutrition is holozoic in animals, that is by ingestion of food. Since there is ingestion of food, we call it as holozoic. So the animal kingdom, they have a definite growth pattern and grows into adults that have a definite shape and size. So the higher forms, they show sensory and neuromotor mechanism. I always say that the head is the most important aspect. High level of cephalization is seen in mammals, isn't it? All our sensory organs are concentrated in the head region with central nervous system, the brain. So head is having all the important components, not only the sense organ but also the central nervous system. And in case of animals, there is higher forms shows sensory and neuromotor mechanism which is not seen in any other animals. Most of them animals are capable of locomotion. The sexual reproduction is by sexual intercourse or mating or copulation of male and female, followed by embryological development. This we see in case of animal kingdom. So copulation and the embryological development. The last part of our discussion in this topic is viruses, viroids, prions and lichens. See, viruses are made up of only two chemicals, but they created a havoc during corona, a multicellular organism like human being. Many of our fellow mates, they lost their lives for this COVID-19 virus, isn't it? So viruses, they are made up of only two chemicals, nucleic acid and proteins. They do not have cell membrane, cell wall. They are acellular. That is why viruses, viroids, prions, lichens, they are not included in the five kingdom classification. So viruses, they are what you call it as, they have the nucleic acid and proteins. So various diseases are caused by viruses. Viruses are a connecting link between living and non-living things. Viroids, they are having only genetic material, free low molecular weight of RNA. Viroids causes a disease in potato, which we call it as potato spindle tuber disease, and it is caused by potato spindle tuber viroid. So prions are infectious proteins. They are just having proteins, infectious proteins. The examples for prions causing a disease is mad cow disease, especially in Britain. That is the reason most of the European countries, they do not import beef from Britain. Reason is they are affected by bovine spongiform encephalitis or mad cow disease. The human analog for this bovine spongiform encephalitis is Creutzfeldt-Jakob disease. Then lichens, we know that they are symbiotic association between algae and fungi. So viruses, they are not mentioned in five kingdom classification. They are non-cellular and not truly living. That is why they are not mentioned in five kingdom classification. So viruses, they have inert crystalline structure. They are obligate, compulsory parasites. They infect a cell. They take over they hijack the cell and utilize the protein synthetic machinery of the host cell to multiply themselves, and they destroy the host cell, and they are going to infect the neighboring cells. So Louis Pasteur gave the name virus, which means venom or poisonous fluid. Virus was first discovered by D.J. Ivanovsky, who was working on tobacco mosaic disease, and he found that it was smaller than bacteria because it could pass through bacterial filters. So he was the first person who discovered virus in tobacco plant. They are smaller than bacteria which pass through the bacteria-proof filters. This is the tobacco leaf. Okay. Beijering demonstrated that the extract of the infected plants could cause infection in healthy plants, and he called it as contagium vivum fluid or infectious living fluid. Then Stanley for the first time crystallized and stored viruses as proteins in the labs. He crystallized and stored them as chemicals in the labs, and the crystals majorly consist of proteins. So virus is nucleoprotein. It has a protein coat capsid and a genetic material which can be either RNA or DNA, but not both. The genetic material is infectious. Now tobacco mosaic virus is RNA virus. Generally viruses that infect plants, this can be asked as match the following. They are single-stranded RNA. So most of the plant viruses are single-stranded RNA. Viruses which infect animals have single or double-stranded RNA or double-stranded DNA. Bacteriophages, they usually have double-stranded DNA. So this is what we have to remember about. They can ask it as match the following, so that point you cannot ignore. So these are the diagrams that they have given in our textbook that you should remembering the labeling. So RNA, in case of this, they have the protein coat is capsid. They are made up of number of units, capsomeres. So RNA is the genetic material. This is a bacteriophage, which has the tadpole shaped structure. The head, neck and tail fibers are there. So they also have a collar. So the tail fibers are there. Sheath is there. So that is a bacteriophage. A virus which infects bacteria, we call it as bacteriophage. A virus which infects animals, zoophage. A virus which infects bird, ornithophage or avian viruses. Then a virus which infects the cyanobacteria, cyanophages. So many such names are there. Whenever we use the term phage, you should understand that we are talking about diseases that are caused by viruses. The protein coat, it is made up of capsomeres. They protect nucleic acid. Capsomeres are arranged in helical or polyhedral geometric forms. So diseases caused by viruses are mumps, smallpox, herpes, influenza, acquired immunodeficiency syndrome, which is caused by human immunodeficiency virus, and that is also an RNA virus. Poliomyelitis is also an RNA virus. In plants, the viral diseases symptoms can be mosaic formation, leaf rolling, and curling, yellowing and vein clearing, dwarfing and stunted growth. So these are all the symptoms of this particular plant viral infections in plants. So viroids, it is an infectious agent with a free low molecular weight of RNA, and it does not have protein coat. It was discovered by Diner in 1971. As I said, even the years you please focus it on. Sometimes they might ask that. These are smaller than viruses. Viroids are smaller than viruses, and it causes potato spindle tuber disease. This is a normal potato affected spindle tuber disease affected potatoes. You can see here the potato spindle tuber affected. This is the viroid particle, that is PSTV. So potato spindle tuber viroid, which is a free low molecular viral RNA without protein coat. We call it as viroids. Prions, these are abnormally folded proteins that causes infectious neurological diseases. These are similar in size to viruses. Viroids were smaller than viruses, but prions are similar to size to viruses. They cause BSE, bovine spongiform encephalopathy, or mad cow disease in cattle, and its human analogous variant, we call it as Creutzfeldt-Jakob disease. In humans, we call it as Creutzfeldt-Jakob disease. The lichens, they have symbiotic association, mutually associated between algae and fungi. Algal component, we call it as phycobiont, and it is autotrophic. Fungal component is mycobiont, and they are heterotrophic. So there are three types of lichens: crustose, foliose, they are leaf-like, and fruticose lichens. Crustose, you cannot peel them. On the boulders and rocks you can find them. They are pioneers of xerophytic succession lichens, you know about them. Foliose, they can be peeled. They are leaf-like. Fruticose, they would be having large fruiting bodies. So these are the types of them. So phycobiont is autotrophic. They synthesize carbohydrates. Fungi, they supply them with water and mineral nutrients. Algae prepare food for fungi, and fungi provide shelter and absorb mineral nutrients and water for its partner. Lichens are, assertion and reason they can ask, lichens are indicators of pollution. Their absence indicates the air is polluted. Both A and R are correct, and R is the correct explanation for A. Wherever air pollution is there, you don't find lichens growing there. So the absence of lichens is an indication for pollution. So they do not grow in polluted areas. So these are the other places where you can see all the videos of my classes. Okay, thank you all. So the MCQ part, I'll also be sharing it with 20 questions solved. Thank you.